Okay, so we're going to continue from last time looking at molecular biology, and last time we were talking about DNA replication. And we looked at this figure to see some of the um, different functions that take place on a chromosome. Right now, I want to focus on the origin of replication, and it's pretty well characterized in bacteria like E. coli, and this is a diagram of the E. coli chromosome showing the origin of replication at the bottom. And again, just like in the other uh, replication origin, once it fires, the polymerases move in both directions, forming this replication fork. Now, in bacteria like E. coli, there's actually a termination site on the other side of the circle. And so the two replication forks don't just simply go around till they meet. If one of them gets there first, it stops, and the other one then will catch up to it. Now, in E. coli, if you grow it under ideal conditions, it can double about every 20 minutes. However, it takes 40 minutes or longer for the polymerase to work its way around from the origin to the termination site. So, in order to be able to replicate the chromosome every 20 minutes, it has to fire that origin before the last round of replication finishes. So that way you can have nested replication forks. So several rounds of replication can be happening on the chromosome at the same time. Of course, if that were to be the case in eukaryotic chromosomes, it would create a mess that would be difficult to sort out. So generally, that is not what happens. And if the origin fires more than once during the S phase or synthesis phase of DNA, it's probably going to lead to some kind of alteration in the chromosome. So let's look at the origin. And what we find here is over a short distance, about a little less than 250 bases, a series of repeated sequences. On the left are code L, M, and R are 13 base pair repeats, and then there are 4 9 base pair repeats scattered out throughout the right-hand side. And this is the sequence that we find at these repeated locations. If you look at it closely, you'll see not only is there GATC, but there's N as well. And what that means is there's no particular base that goes in there. N stands for any nucleotides. So those are not conserved base positions. Okay, so the first thing that's going to happen is a protein called DNA-A, for lack of a better name, I guess, bind to the 9 base pair repeats, and that's shown in the middle diagram on this slide. So you can see 4 molecules of the DNA-A binding to the 4 9 base pair repeats. Once that happens, then many more of these molecules can bind to the protein as well as to the DNA, causing it to loop around as shown in the bottom. And basically what that does, it twists the DNA and allows the 2 strands to open up. Now you might notice on the previous slide that these 9 base pair repeats are AT rich, again, which make them a little weaker in terms of base pairing so that the 2 strands are a little easier to separate. So once we start to separate those strands, then other proteins, DNA B and DNA C, come in and start to pull those two strands apart even more. And once that happens, then the helicase can come in and separate the strands and start moving the replication forks in opposite directions. So we have this origin of replication shown at the top here. These initiator proteins come in, bind to these repeated sequences, twisting the DNA, which starts to pull the strands apart. This allows more proteins to come in and pull them apart even farther, so the helicase can get in and start to separate the two strands. Then the primase can come in and make an RNA primer, which then can be used to elongate off of with DNA polymerase, and so the replication starts. Now, in eukaryotes in particular, but I suppose in bacteria as well, we need to know which strand has been replicated and what hasn't. And one way of telling that is by this method of methylating the bases. And so if DNA is methylated, it's identified as old DNA, and the unmethylated DNA as a newly replicated strand. So you can see here, if you start out with DNA shown on the left, and there's a little four-base sequence here, GATC, it's methylated at the A. And you'll notice if you look at the top strand in the 5' to 3' direction, it's GATC. If you look at the bottom strand and you go 5' to 3', which would be the opposite direction now, going from right to left, it's still GATC. So those are the same sequences on both strands. So they're complementary replicated or methylated here. This allows the methylation enzyme to identify this GATC if one strand is methylated and methylate the other side. So during replication, of course, we use the methylated strands, DNA, as a template, synthesize new DNA, which will not be methylated. So again, at that point, we can tell the difference between unreplicated and replicated DNA, or newly replicated DNA. And then the methylase comes in and remethylates the new strand to match the old one. So again, this shows original DNA duplex methylated on both strands. Two strands are separated, used as templates to make two new strands. Those will initially be unmethylated, and then the DAM methylase comes in and methylates the new strand. Now, unlike bacteria, eukaryotic origins are kind of poorly understood. And the first one that was identified was in a yeast, a Saccharomyces extrachromosomal piece of DNA called a 2-micron circle. So this is a circular piece of DNA, like a plasmid in bacteria, that's found in yeast, and it's called a 2-micron circle because it looks like it's about 2 microns when you see it in an electron micrograph. So there's a region of short sequence in the 2-micron circle shown here, and I believe it's about 11 bases long. Yeah. And the top here shows how important each one of these bases are for the two micron circle to be replicated. Okay, so what was known for quite a while was that if you had this 11 base pair sequences on the piece of DNA, it would be replicated. If you didn't, if you removed it, it would no longer replicate. You could take this small fragment, insert it into another piece, put it back into the yeast, and it would be replicated. Now that does not mean it's an origin of replication. It means it's a sequence that directs a replication. So it was termed an autonomously replicating sequence, or ARS. It was subsequently shown to be a replicating origin by some very detailed and technical experiments. I'm not going to go into, but some very elegant work. Now, as I mentioned earlier, in eukaryotes there are multiple origins of replication, and this diagram shows that. Now, when you have multiple origins on a chromosome, they can be at various distances apart. They can initiate replication at different times, and it's been shown that actively transcribed DNA seems to be replicated earlier in the process than inactive, as a general rule. But when two origins that are near each other initiate synthesis and the replication forks move out in opposite direction, at some point they're going to meet. And when that happens, the two replication bubbles fuse into one big one, and then the forks on the outsides keep migrating until they hit another place where there's a replication fork. And you can see this in electron micrograph here, the replication bubbles. One down at the bottom here is about to fuse with another replication bubble. Now, it's also known that in some organisms, like, again, insects, there are many more active replication origins in early development than later on. And that's essential because in Drosophila, the nuclei post-fertilization are dividing every few minutes, and so you have to have a large number of replication forks in order to synthesize DNA that fast. Later on in development, that's not the case. Cells are dividing much more slowly. You don't need as many replication forks. Now, one problem that arises when replicating linear chromosomes is, what do you do at the ends? Because as I mentioned, the polymerase can only go in the 5' to 3' direction. So on the one strand, it can't go to the end. Also, we need a primer, and so it has to synthesize a small piece of RNA, and there's no way to, well, you can get rid of the RNA, but you can't fill in with the polymerase. So there's a special mechanism for synthesizing the ends of eukaryotic chromosomes, and that's putting these telomeres on the end. And so what's used is this enzyme telomerase, which is a combination of RNA and protein. So the RNA acts as a template to synthesize DNA off of. And so it can synthesize a short region of DNA and then back up and do it again and put this same sequence on the end of the chromosome over and over again. So you get these repeated sequences on the end of eukaryotic chromosomes. And different species have slightly different repeated sequences. So these telomeres now are found at the end of of all eukaryotic chromosomes, and they need to stabilize the end of the chromosome. Now, eukaryotic, prokaryotes the same way, cells do not let ends of DNA just sit there. So if you have a fragment of DNA, or if the cell finds that there's a fragment of DNA, it's going to do one or two things. It's going to either chew it up, or it's going to attach it to another piece of DNA. And sometimes it does some of each. It'll chew back on it and then attach it. So in order for that not to happen, we have to have a special configuration at the end of these chromosomes so they don't get either chewed up, or we don't attach all the chromosomes to each other and make one giant chromosome. And this figure shows kind of how that does it. It loops back on itself and forms this structure shown in the circle here, which protects the end from nuclease digestion. And there are a number of proteins shown here that are used to stabilize this structure so that it doesn't get chewed up. Now, you may have heard a lot about these telomeres in the last several years. There's been a lot of work done looking at the length of telomeres and how they shorten with age. And so that cells become part of the senescence process of cells is the shortening of these telomeres. In other words, having fewer and fewer of the repeated sequences. Okay, before we move on, I just want to review some of the major points for DNA replication. Again, DNA polymerase cannot initiate synthesis. It requires a primer to elongate off of. Replicating DNA requires a template following the AT and GC base pairing rules. The polymerases can only synthesize in the 5 to 3 prime direction. They cannot go backwards. Mistakes during polymerization are minimized by proofreading. That is the 3 prime to 5 prime exonuclease. So if a wrong base gets placed into the nascent DNA chain, the polymerase can monitor that, can see if it's the right one or not. If it is the wrong one, it can chew it out with that nuclease and then insert another one, trying to get it right the second time. So once synthesis is initiated at an origin of replication, the 2 replication forks will move away from the origin in opposite directions. Okay, so the lagging strand must be synthesized in short Okazaki fragments, and each of those have to start with an RNA primer. And so the lagging strand, of course, is a strand where the polymerase is moving away from the fork rather than towards the fork. After eukaryotic chromosomes are replicated, then telomeres are added to the ends of the chromosomes, these repeated sequences, and then they are stabilized by looping back, and then a large number of proteins are involved in holding those in place and stabilizing it. Okay, let's go back to this diagram. We've talked about replication. Now we're going to talk a little bit about how DNA changes over time. It does so pretty slowly. DNA is an incredibly stable molecule, but it does change, and how the cell minimizes that change by repairing any lesions or insults to the DNA. And then we'll talk a little bit about recombination. Now, before we talk about the types of changes that we get, I just want to mention that we looked at the difference between the structure of RNA and DNA and that the the difference is at the 2' position on the ribose, on the sugar, where there's a hydroxyl group on the RNA and just a hydrogen on the DNA. And this makes a huge difference in the properties of these molecules because the deoxy form of the molecule is extremely stable. So stable that we can pull DNA out of animals that have been dead for centuries and still get some DNA out of it. Whereas RNA turns over very rapidly, it very quickly disappears. And the reason for that is that hydroxyl group, the hydrogen on that hydroxyl group is actually an acidic proton. And so the higher the pH, the more likely that hydrogen is to come off the oxygen, leaving a negatively charged oxygen there, which attacks the phosphodiester bond and breaks it. And that's why if you have a mixture of RNA and DNA and you want just DNA, you can get rid of your RNA by adding base, and it will chew the RNA up and leave the DNA. The other thing that it's allowed is the nucleases. The ribonucleases are much simpler molecules and much more stable than the deoxyribonucleases or DNases. In fact, in the laboratory, what we do to get rid of DNase, if we want to have ribonuclease, is to heat the protein up. It destroys the DNase, but as it cools back down, the RNase reforms and is active again. Okay, so DNA is a very stable molecule, but it does change over time. And these, again, we've mentioned this before, but look at it again. These are some of the ways it changes. Single bases can change to make a mutation. We can get duplications of regions of the genome, and that's happening all the time. And when we looked at the content of the human genome, we saw that few percent of the genome is made up of these duplications, and they can be different regions of the genome that are duplicated from one person to another. We can also get what's called here DNA segment shuffling. So parts of genes can be switched with parts of the gene from other chromosomes, which can bring in new activities. And then the last way we can acquire new DNA is through horizontal transfer. So even though we have mechanisms that identify foreign DNA and try to get rid of it, it's not 100% fallible. So sometimes foreign DNA does get into an organism and get incorporated into the genome and is therefore stabilized. Now there's two ways that homologous genes can drift apart, and the one shown on the left here is when you have a gene, an ancestral gene, the organism replicates, and the two daughter cells or organisms follow different lineages, so they start to diverge over time. The other possibility is that the gene can be duplicated in the same cell, and then those two genes can diverge over time and potentially create new activities. Okay, I want to now talk about DNA rearrangements, and we're going to start talking about recombination. Then we'll talk about gene amplification, transposable elements, and then finally mutagenesis and DNA repair. So when we talk about recombination, we're simply simple definition is the reciprocal exchange of genetic information between two chromosomes. And the key word is reciprocal because a homologous recombination involves the exchange of the same information. So just shuffling the DNA between the different chromosomes. So no information in this case would be gained or lost by either of the chromosomes. So the next simple figure shows how that would work. So in this case, on the left we have the blue chromosome and a black chromosome. After a recombination event between sister chromatids, we're left with two hybrid chromosomes, half blue and half black, and then one half black and half blue. But we have not gained or lost any information on those two chromosomes, so they all still have the A, B, C, and D regions. Now we can have reciprocal recombination between repeated sequences on the same chromosome. So if they are direct repeats, meaning they're in the same orientation, they can loop around, pair up based on the homology. And if we get a crossing over event between those two, the result is looping out of the region in between. So we're left with one copy of the repeated sequences, which is a hybrid of the two repeats, depending on where the crossover occurred, and then a circle containing the other repeated sequence. Now this can happen in the reverse direction. If there's a circle of DNA that contains a region that's homologous to the chromosome, it can recombine into that chromosome and producing two repeats on either side of it. Now if we have homologous sequences that are in opposite orientation, so they're inverted repeats, they can loop around and pair as shown in the middle of this figure. And when there's a recombination between those, we don't lose anything, we just invert the region between the two repeated sequences. So at the top, before the recombination, the orientation was A on the left, B on the right. After recombination, B is on the left and A is on the right. Okay, so I want to talk first about a little bit about the mechanics of recombination, how this takes place. Then we'll talk about different types of recombination, a little different variant on recombination called gene conversion. Then we'll talk about unequal crossing over, where DNA is gained and lost on the two homologs. And finally, these transposable elements that can move around in the genome. Okay, so let's look just briefly at the mechanism of recombination. Okay, so a nick in one strand allows invasion of the homologous double helix. Nick is what we call when one of the strands has a break in it but not the other. Okay, so if we have two homologous chromosomes, a green and a red here, and we get a nick in this case in the green chromosome, that now those strands can separate, and that green strand hanging off by itself then can invade the other homologous chromosome, pushing off the homologous strand and binding to the complementary strand. Then if we get a break in the other chromosome, it can, since it's floating around now, it can then pair to the single-stranded region on the first chromosome and form this structure shown on the right here. Then if we ligate those together, now these two chromosomes are attached. Now, single-stranded NICs can initiate this process, but actually double-stranded breaks initiate recombination at about 10 times the rate as single-stranded. So once we have this configuration shown on the right, which is called the Holliday junction, and we can depict it in this next picture. And so we see it again up on the top, the Holliday structure or junction. And at the bottom, this is exactly the same configuration. It's just been twisted to put in the plane of the page here. So we see up in the top left the two blue strands with the dark blue attached to the red one. Down in the bottom left, the original red and orange strand. Across from that is the original two blue strands, and then at the bottom right, the red and orange with the dark red attached to the dark blue here. Now, at some point, we need to resolve this and separate the two chromosomes back again. And the way it resolves will determine what the outcome is, whether there was recombination and where it occurred, because this Holliday junction then, after it's formed, can move up and down the chromosome. And where it's resolved, when it's resolved, where it is when it's resolved, can make some difference. Now again, a double-stranded break can initiate recombination about 10-fold more than single-stranded breaks. And so what's shown here is, again, we've got two homologous chromosomes at the top, double-stranded break in the red one at the bottom one. Then, as I mentioned before, if you have ends of DNA, they're either going to be ligated to something or they're going to be chewed back. So if they're chewed back by a nuclease, we get a gap in this chromosome. Okay, so then when in this case the dark red strand invades the blue chromosome, it can be used then as a primer to elongate off of with the DNA polymerase that can displace, in this case, the blue strand. So as it keeps going, as the polymerase keeps going down the strand, displacing more and more of the blue strand, then that strand can come over and hybridize to the red chromosome. So again, then we need to resolve these Holliday structures. There'll be two of them here, and they can be resolved in two ways. One way puts it back the way it was, and the other exchanges the information on the two chromosomes. So recombination then can result from a number of different things, from due to reciprocal exchange during meiosis or mitosis, can be the result of DNA damage, can be the result of introduction of foreign DNA, and some viruses, particularly bacteriophage, have regions on their genomes that are homologous to regions in the bacterial chromosome, and so can insert themselves into the bacterial chromosome by homologous recombination. We'll also can see that some cases during development there are programmed recombination, which results in the generation of new genes, basically, and we'll show you an example of that. Okay, so we've talked about this or alluded to this a little bit, this reciprocal exchange during uh, meiosis or mitosis. And this again is a condensed chromosome with two sister chromatids after replication before the two are pulled apart and go into separate daughter cells. If we zoom in, what we see are the two sister chromatids separated by this central element. Now again, a simple reciprocal exchange between sister chromatids is going to result in exchanging part of, in this diagram, the blue chromosome with the black chromosome, but again, everything is still, everybody gets some of each. So even though the top chromosome is now half blue, half black, it still has all four of the segments shown here, A, B, C, and D, and the same with the bottom chromosome. So we can label one of the strands with a dye that fluoresces so we can tell it apart from the other strand, and then we can look in the microscope, fluorescent microscope, during mitosis, and you can see where recombination has occurred. One place is, you can see on many of the chromosomes there's been a crossing over. One that's easy to see is right in the middle of the field here, the chromosome that's pretty much horizontal. You can see there were two recombination events, so that the fluorescing chromosome starts out at the bottom on the left, then it flips to the top, and then comes back to the bottom again. And if you look at some of the other chromosomes, you can see that in any given round of cell division, there are a number of sister chromatid exchanges taking place. This is an example of an abnormal level of sister chromatid exchange, where you can see there are many events along each chromosome. And this is taken from a cell from a person with a syndrome called Bloom syndrome. And it turns out the defect in Bloom syndrome is in the DNA ligase, the enzyme that links the Okazaki fragments together. And so if you don't ligate those fragments together, you've got a bunch of single-stranded nicks that can promote recombination during mitosis. And so the result is this high level of exchange between sister chromatids. Now we can also get crossing over between non-sister chromatids during meiosis, and these are still homologous chromosomes, but the same type of event occurs. And so we can get shuffling of the genes between our homologous pairs of chromosomes during mitosis as well, but also in meiosis. Okay, I want to talk a little bit now about DNA damage. One of the things that causes double-stranded breaks in DNA is X-irradiation, and this is something that you'll all be familiar with because dentists use X-rays on a regular basis. And so these X-rays can break the DNA, cause double-stranded breaks, and induce recombination. And this is the same figure we looked at a few minutes ago. I'm not going to go back into detail, but again, showing you if you have this double-stranded break, then the strand that's broken can invade homologous chromosome, where it can be used as a primer to elongate off of and fill in these gaps. And Okay, we can also get introduction of foreign DNA into cells by recombination. And I'm going to show an example here. The life cycle of the bacteriophage lambda, which is a virus that infects the bacteria E. coli. So at the top we have a depiction of the bacterial cell with its circular chromosome. The phage then attaches to the surface of the cell, injects the DNA into the cell as a linear molecule. It circularizes, and then it can go through two processes. It can go through the lysogenic process shown on the left, where it inserts itself into the host chromosome by homologous recombination. And for this purpose, we can think of the E. coli chromosome as linear because it's very big. And if we have a circle and a linear piece of DNA that have a region of homology, they can pair up. We can get recombination at that site, and the circle is inserted into the linear piece of DNA. And remember, now there are repeated sequences on either end of the inserted piece of DNA. So the lambda can come in, insert itself into the E. coli chromosome, and stay there for an indefinite period of time. So every time the E. coli replicates itself, it replicates the lambda along with it and carries the viral DNA along with its own DNA in the E. coli chromosome. Now, if the lambda senses that the cell, the E. coli cell, is not doing very well, and it has a number of proteins that are used to sense if the E. coli is happy, if it's replicating normally, and if it senses that that's not the case, it will reverse itself. It will recombine out of the host chromosome and go through the lytic pathway where it replicates itself, makes all the proteins that are needed for packaging the viral DNA into a viral particle, and then it will lyse the E. coli cell, releasing viruses that will go out into the world and find a new host. So again, the insertion of the lambda DNA into the bacterial chromosome occurs through a homologous recombination event where there's a sequence on the lambda that's homologous to a region on the E. coli chromosome. There's also a protein coded for by the lambda, which we call integrase, which facilitates the recombination that inserts the viral DNA into the chromosome. Now, non-viral DNA can enter the cell and become inserted into the host chromosome by homologous recombination as well. Again, usually those aren't going to carry an integrase with them, but it still can happen. And of course, it's a much less frequent event, but it's still occurs on a regular basis. Okay, I want to talk a little bit about programmed recombination, and this is the most well-characterized example of that, although there are other examples, particularly in the olfactory system. But this is in the immune system, and what we have for antibody-producing genes is this configuration on the genome, where there are a number of what are called V regions, standing for variable. And then, so that can be a couple hundred maybe. Then there are a few of what we call the J regions, shown in the little green bars. These are J for joining. And finally, the green bar, the C region, the constant region. So in developing B cells, these rearrange to take one of the V regions, put it next to one of the J regions, and then it's transcribed through the C region, and the region between the J and the C is spliced out to give this mature RNA. So in this way, we can generate a lot of different antibody molecules, antibody producing cells, um, by having these different variable regions that are attached to these other two regions. So these rearrangements occur randomly in developing B cells, but once they make one that identifies an antigen, then that cell proliferates, divides several times to make a lot of cells that all produce the same antibody. So each cell produces just a single type of antibody, and it's made up of one of the V regions, one of the J regions, and a C region, as the result of a programmed recombination in the developing B cells. Now, as opposed to recombination, which is the reciprocal exchange of genetic information, gene conversion is the non-reciprocal exchange of genetic information. And I'll show you how that works on the similar diagram that we looked at for recombination. Again, we have the blue and the black chromosomes. They have 4 regions, A, B, C, D on them. In a gene conversion event, one of the chromosomes will not change, and this is shown by the blue chromosome, still A, B, C, D, it's all blue. But the other chromosome now has acquired information from the blue chromosome and exchanged that for the original information on the black chromosome. So now we've duplicated that region of the blue chromosome onto the black chromosome, and what was originally there then would be lost. It's difficult to study gene conversion because you have to be able to follow all 4 of the gametes that are derived from one mitosis. And as I mentioned before, in most organisms, we only get one of those out. So gene conversion has been more thoroughly studied in yeast, where all 4 of them lead to new progeny, whereas in other organisms we can just kind of try to piece together what's going on based on just seeing one of the results of meiosis, one of the 4 results. Okay, I want to talk now a little bit about unequal crossing over, and as the name implies, in this case we do not get perfect lining up of the chromosomes so that we get equal exchange. And let's look at a diagram here. If you have families of tandemly repeated genes, they can pair up differently, and so we can get the gain or loss of copies due to this unequal crossing over. So what's shown here in the top figure is 2 copies on the red and the blue chromosomes, and if you get pairing of the first copy of the red with the second copy of the blue, the result will be on one chromosome 3 copies, and on the other chromosome 1 copy. And so on the top chromosome we get one of the blue copies back, one of the red, and then a hybrid of the blue and the red. And then of course on the other chromosome we get 1 copy that's half red and half blue. Now this happens at a fairly frequent level in repeated in these gene families where there are repeated sequences that are fairly homologous, and this is an example of that in the beta globin gene cluster. And what's shown at the top, the blue arrows show the different genes in the beta globin cluster. The beta globin gene on the left, or I'm sorry, on the right, far right, the delta globin, which is a beta globin that's expressed in the fetus. We have to have a slightly different hemoglobin in the fetus in order to exchange oxygen between the mother and the fetus. So the fetus globin is slightly different. To the left of that is what's called a pseudogene. This is a region that's homologous to the beta globin gene, but clearly not being expressed at the time. And then there are other genes to the left of that that are, again, fetal versions of the globin gene. So a number of deletions in the beta globin gene cluster have been identified. That, of course, leads to some issues, some medical issues called thalassemias. And these bars underneath the blue bars depict the areas that have been deleted. So the pink represents what's still there, what's remaining. The white part of the bar shows you different deletions that have been identified. And you can see that these are due to, again, deletions within one of the beta globin-like genes, between one of the beta globin-like genes and another beta-like genes, presumably due to this unequal crossing over. We can also find examples where the reciprocal of this is so that there are extra copies in the beta globin gene cluster. Those are harder to find because they don't really present any medical issues. So the people with the deletions are much easier to find because they have these thalassemias. We also see this in the alpha globin gene cluster, the same idea here. These are the blue sequences at the top are the alpha globin genes. Again, a number of pseudogenes that are shown in just a bar with no arrow since they're not expressed. But we see, again, a number of different thalassemias that are derived by unequal crossing over between these genes. Now, if we have a tandem array of genes, they can be kept very similar by a combination of unequal crossing over and gene conversion. So if we have a mutation in one of the genes, as shown in the top left of the figure with a little red line, we can one of two things is going to happen over time. Either that particular mutation is going to repopulate the chromosome so that all of the copies of the gene match that, or it's going to be deleted. And through a series of unequal crossing overs, one or the other of these is going to happen. If you have 10 copies of the gene, your chances of the mutation establishing itself is probably about 1 in 10. And on the right, we see a gene conversion event that, again, can help repopulate the genes with a new mutation. Of course, the opposite can happen, and we can correct that mutation back to the original. sequence. Okay, so we can also get this gene amplification, which we've mentioned a couple times. And this is when the cell specifically increases the copy number of a particular gene or region to a greater extent than it increases copy number in the remaining genome. And when we looked at the schematic of the human genome, we saw that this little purple region between 45 and 48% depicts the amount of DNA that might be duplicated at any given time in a genome. Now we can select cells that have many copies of a gene, and this shows how this was originally done by growing the cells in the presence of a drug that inhibits the protein. In this case, the drug methotrexate, which we talked about earlier, that inhibits the enzyme dihydrofolate reductase, which is needed for DNA replication. If you grow cells in the presence of methotrexate, only those cells that have extra copies of the DNA can grow. And I should mention there are other ways that you can overcome the drug or develop resistance to the drug by mutating the enzyme. But one way of doing that is to make extra copies of the DNA. And again, I want to stress that this is a random event. We're not causing the cell to make extra copies. We're selecting out those that have made extra copies of the dihydrofolate reductase gene. But if you select out cells that can grow in methotrexate, you can then stepwise increase the concentration of that drug and select cells that have more and more copies of the gene until you can get cells that have 1,000 copies of the dihydrofolate reductase gene instead of the normal 2. Now, when you look at the chromosomes of these cells that have all this extra DNA that codes for the dihydrofolate reductase gene, you find 2 different types of DNA here. On the left are these small extra chromosomal elements, these little yellow dots, which are called double minute chromosomes. And they're called double minutes because, again, this is a mitotic spread. This is actually a mouse cell. And so these extra chromosomal elements are replicated, and they are paired just like a chromosome before mitosis. Now, they don't attach to the spindle and segregate or assort perfectly like normal chromosomes do, but they are passed on to the daughter cells. Other cells that have acquired this extra DNA sometimes attach them all together and insert them into a chromosome. And that's what we're seeing on the right here, this big yellow area, and that's due to probing it with a fluorescent probe to the dihydrofolate reductase gene. And this is sometimes called a homogeneously staining region because unlike the rest of the genome that forms this consistent banding pattern when you use a dye like Giemsa, the homogeneously staining region, or HSR, does not show any kind of banding pattern. Now, one difference in the property between these 2 types of cells is that cells with the homogeneously staining region, those that have incorporated the DNA into the chromosome, are stable. In other words, if you take the drug away, the extra DNA does not go away very rapidly. Unlike with the double minutes. If you take the drug away, you rapidly lose the extra copies of the DHFR gene. And that's because when the cells divide, they don't assort perfectly. So some cells get more copies of more of these double minutes, and some daughter cells get less. And those that get less have an advantage in time it takes to replicate, and so they quickly overtake the ones that got more. Now there are a number of examples of gene amplification during development, and this is one example, again in the fruit fly, Drosophila, and this is a region of the chromosome that codes for eggshell proteins or chorion proteins. Insect egg shells are made up of protein, unlike chicken eggs, which are made of calcium. And so at a late stage in the development of a fly egg, a lot of this chorion protein has to be produced in a short time. And in order to do that, the fly amplifies up the region of the chromosome that contains the chorion protein genes. And this is shown in this figure. If you ask how much DNA there is, how many copies there are over this region of the chromosome, you see this bell-shaped curve shown in the middle figure here in figure B. And the chorion genes are at the peak of the curve. Now you can look in the electron micrograph and see these nested set of replication forks. So there's an origin of replication near these chorion protein genes that is firing several times to produce this series of nested replication forks to produce this amplified set of genes in the follicle cells that surround the developing egg. Other insects, like the silk moth, just have many copies of the chorion genes in their genomes, and they replicate them every time the cell divides. But insects like Drosophila only carry one copy of each gene and just amplify it up when they need to make a lot of it. Again, there are other examples of this in other insects, and as well as in frogs. Not aware of any examples in humans, but that doesn't mean there aren't any. Okay, we're going to move on now and talk about transposable elements. Again, these are pieces of DNA that can move around in the genome from one place to another. And there are three types of elements. What, for lack of a better name, at the top of this table is called a DNA-only transposon. Then there are the retroviral-like transposons that go through an RNA intermediate, and then the non-retroviral retrotransposons. And we're going to talk about the first two here. So this is the structure of the transposase shown at the top. You see a piece of DNA is depicted by these two orange lines. On either end of the transposase are repeated structures, and they're inverted, so they're inverted repeats. In the middle of the transposase is a coding region that codes for a protein that facilitates the integration or the excision of the transposase, so it moves the transposable element from one location to another in the genome. And that's simply called transposase. Keep in mind, though, that some transposable elements don't have a good copy of the transposase, and you don't need that if there are other elements in the cell that have a good gene, because it can make the protein, and the protein then can act on these what we call incomplete transposable elements. Okay, so that gene acts in, we say, acts in trans as opposed to cis. And I think Dr. Bell will talk more about trans and cis ideas in regulating gene expression. But it's the same as you learned in organic chemistry. It's together or opposite. So basically, a cis element is one that is present and functions on the DNA. So in this case, the inverted repeats would be cis elements. Trans element usually acts through a protein like the transposase. Okay, so again, you don't need a good transposase in the element that you're moving. You just need that gene somewhere in the genome so it can be expressed. So when the element is integrated into the host genome, it duplicates a short region of the host chromosome. And this is shown in the middle of the double blue line. And a small region, in this case of 5 bases, is duplicated when the transposon is integrated into it. So what you find when there's a transposable element in the chromosome is a region of DNA, often containing a coding region, and that codes for transposase. And then inverted repeats, and then outside of those, direct repeats. And so there's a little table here at the bottom of this figure showing some different transposons, sometimes called insertion sequences, and that's the IS in the names of these. And you see that there's a variation in the target repeat as well as in the inverted repeat. And so it can range, the duplicated region can range from 5 bases up to 12 or 13. Inverted repeat is usually a little longer than that. It can be as long as 40 bases. Now, anytime enzyme works on DNA, there's going to be some specificity, even though we refer to a lot of enzymes as random. They're still going to have some preferences, and some of these transposases are quite random, whereas others are a little less random. And what it's shown on the right side of this table is the specificity or lack of for some of these different IS elements. Again, IS elements, insertion sequences, transposons or transposable elements all mean the same thing. Okay, so when a transposable element inserts itself into the genome, it makes a staggered cut in that genome, as shown in this figure. So it cuts the 2 strands at different locations. Then it separates them, inserts the element into the target site, and then leaving 2 single-stranded areas on either end, on opposite strands on either end, and then those are filled in by the repair enzymes, the DNA polymerases that repair DNA. And of course, they can be ligated in with DNA ligase. So you get this duplication when these elements are inserted into the genome. Okay, so these transposable elements then can be excised from one location and moved to another. Some of them appear to be pretty random. Others appear to be have some kind of control. But a typical transposable element seems to move somewhat randomly. So when you have this element in the chromosome, it can be excised using the transposase, and then it can go to another location in the chromosome and insert itself, again making this staggered cut, uh, inserting itself. Then the host enzymes come in, fill in the gaps, and generate this short direct repeated sequence. So with this event, there's no gain or loss of DNA other than the short repeated sequence. Now when the element is excised out of the chromosome, it leaves those two direct repeats in there. So it doesn't revert back exactly the way it was. There's a little piece of added DNA that's repeated twice there. Now, most of the time when a transposable element moves, it moves pretty precisely, but sometimes it doesn't. It's a little less precise, and this shows some of the rearrangements that we see due to transposable element. And so in this example, we've got this chromosome shown in green, and it's labeled, regions on it are labeled 1 through 9. Transposable element, red, is labeled A through F. Okay, now it inserts into the chromosome, duplicating a short region is shown as regions 5 and 6 on the chromosome. So you can see there's a 5 and a 6 on either side of the red element. And now when it excises, again, most of the time it's going to be pretty precise, but sometimes it leaves part of the element behind. That's what's shown on the left at the bottom. Okay. Next to that is a precise excision, leaving this short repeated sequence. Sometimes it can leave a little extra DNA, shown in the third chromosome on the bottom, or sometimes it can take some of the host DNA along with it. And so in this way, a transposable element can move pieces of DNA around from one place in the genome to another. It can also disrupt genes. It can, if they're, can change the expression of certain genes. So it can have some notable effects if it inserts itself into the right position. Okay, the second type of transposable element that we're going to talk about are called retrotransposons. And they have a similar life cycle to a retrovirus. And we've probably all heard of retroviruses. The AIDS virus is a retrovirus, fairly complicated one, but has the same life cycle. But this is the life cycle of a retrovirus. Starting at the left, we see a retroviral viral particle enters into the cell, releases the RNA, and this is an RNA virus, so it packages RNA for its genetic material, not DNA. But the first thing it needs to do when it gets into a cell is convert that RNA back into DNA using the enzyme reverse transcriptase. And so that reverse transcriptase is RNA-dependent DNA polymerase. In other words, it uses RNA as a template to make DNA. Once it's done that, it can take that DNA strand shown in red and use it as a template to make the other strand. So now we have double-stranded RNA. At this point, it can be integrated into the host chromosome. Okay? And once it's in there, it then can make new RNA transcripts, shown over here on the right in the blue lines. Those can then make the proteins needed to package it, as well as can be used as the genetic material to be packaged. And so can make new viral particles that then can go out and infect other cells. Okay, a retrotransposon has the same life cycle, except it doesn't package the RNA into a viral particle and send it out to other cells, but it can make itself into an RNA molecule, reverse transcribe that back into DNA, and insert it back into another location. So unlike the previous type of transposable elements that we talked about, where it moves around but it doesn't increase in copy number, every time the the retrotransposon inserts itself in a new location, it's an additional location, and the element is still present in the previous location. So in this way, retrotransposons tend to accumulate more and more over time in a cell. Some organisms, some cells have a huge fraction of their genomes are retrotransposons. And in fact, these lines and signs that we see in humans are retrotransposon-like elements. Okay, I want to talk about mutagenesis now, and so how the genome can change over time. And of course, we mentioned a little bit that there certainly are errors during replication. They're not very frequent, but they do occur. Many of them can be corrected, but not all of them. We also have injury to DNA that causes a change in the base structure. And that can be due to chemical insult or to radiation, either ultraviolet radiation or X radiation, which we've mentioned previously, and we'll talk about a little more. And again, all these gene sequences are changing over time, but different proteins evolve at different rate. And here are 3 examples looking over a timeline on the x-axis and the amount of change on the y-axis. And we see the blue line represents changes of protein cytochrome C, which is involved in metabolizing foreign chemicals or drugs. And it's evolving very slowly. Hemoglobin in the middle, the red line, is evolving at a little faster rate. And the third example, fibrinopeptides are evolving very quickly. And I mentioned, I think, previously the fibrillin genes on oral bacteria, Porphyromonas gingivalis, evolve so fast that even fairly closely related strains have these genes with very little homology. They do have regions in the gene that are very homologous, but other regions, the region that's presented to the outside of the cell, has very little homology because it's changing very rapidly to try to fool the host into thinking it's not there. Okay, again, there's 2 types of mutations: base substitutions, or what we call point mutations, and DNA rearrangements. And you can have many different types of DNA rearrangements. Some of them shown here, deletions, where just simply part of the chromosome is missing. Can have duplications, can have so that there's extra DNA present that's the same as another part of the chromosome. Now in this case it's showing it right adjacent to it, but it doesn't necessarily have to be. You can also find inversions where part of the chromosome has been flipped around and reinserted into the DNA, and translocations where we have exchange again between two different chromosomes. And more often than not, this is a homologous event, so there's nothing gained or lost. But there are also times when we have unequal crossing over, so one chromosome gains DNA and one loses it. Or we can have a gene conversion event where one chromosome has donated its information to the other one, but maintains the structure that was originally there. Now, X radiation can cause these double-stranded breaks in DNA, and again, this induces recombination. It also can induce cause loss of DNA by nuclease digestion off the ends of them. So this is a chart showing the annual exposure to X-rays, and you see most of that is due to natural radiation, but there's other sources as well as diagnostic sources. And what I want to show you here is a graph showing the amount of change in a chromosome over different doses of X-rays. And you see this is a linear relationship. So the more X-rays you're exposed to, the more damage to your DNA, or the more likely there is damage to your DNA. And you can see that line goes right back to zero. So as soon as we start to get exposure to X-rays, we are exposing ourselves to any kind of mutations, particularly double-stranded breaks, which can cause either loss of DNA or rearrangements in DNA, including recombination. Now, sometimes these rearrangements can bring previously unassociated sequences next to each other, and this is one dramatic example of a rearrangement causing a different activity. In this case, the coding regions for development of the thoracic region in the fly, where the legs and the wings are, is moved next to the regulatory regions that express proteins in the head. The result is a pair of legs growing out of the fly's head where its antenna ought to be. So again, that's a pretty dramatic example, but we can get other more subtle things going on as well. Okay, I want to talk now a little bit about single base substitutions and some of the different things that we see. And I just want to impress upon you the variety and the amount of different insults that can occur in DNA that lead to a change in the base pairing. Though we don't need to memorize these, but one example here is the deamination and depurination. And these are the two most frequent spontaneous chemical reactions of DNA. And they're shown here. The deamination converts the cytosine back to uracil. And so then we have, instead of a C in that place, we have a U, which base pairs just like a T. So if that would happen in the DNA, we would change a CG base pair to a TA base pair. The other thing that we get is depurination, where the purine, in this case, example shown here, the guanine is removed, and then there's no base there. So when DNA polymerase goes through to use this as a template, there'll be no base to put the complement into, so it will just have to pick one. So again, this is showing the conversion of cytosine back to uracil and the changing in the base pairing. At the bottom, if we get a deamination on adenine or adenosine when it's in the DNA, again, this is going to change the base pairing, and so we have hypoxanthine here, which will pair with cytosine better than with thymidine, so that we get a conversion from AT to GC if we get deamination of adenine. Now this figure shows all the different places that we can find alterations that require repair. We don't need to go through these. I just want to again impress upon you that there are a large number of different alterations that can happen. The other issue that we could have is if you're exposed to ultraviolet light, it can form these pyrimidine dimers. So if there are two pyrimidines next to each other on the DNA and they're exposed to ultraviolet radiation, they form a dimer between those two bases. And this can, of course, it can be repaired, but it will disrupt expression. And I'm not sure what happens if you try to replicate through there, but it will certainly disrupt expression or transcription through that gene. In fact, I've used the technique in the lab to identify the beginning of transcription. If you treat cells with UV radiation and then you look at the transcripts along this region, along a given region, you can see that the farther away from the start of the gene, the less number of transcripts there are, because the more likelihood that you've caused a pyrimidine dimer and disrupted transcription. So you can plot that as a straight line right back to the start of the transcription, and so you can map transcription units that way. We did that many years ago with a gene I was studying, and actually found that there was another gene just a few bases away and being transcribed off the other strand, so going in the opposite direction. Before that, we had thought that transcription there was just part of the same gene. Okay, another type of alteration that we get is alkylation of the DNA, and formaldehyde is the alkylating agent, with one carbon. And so again, this can change the base pairing. And this is why, one of the reasons why you want to stay away from formaldehyde. There are a number of other common alkylating agents shown here. Some of these have been used to treat cancer by trying to disrupt, again, DNA replication by alkylating the enzyme, or alkylating the DNA. Okay, if we have a deamination, it can be repaired or it can lead to a mutation. So in the example shown on the left, we have the deamination of a C. Then that base can be removed and it can be replaced with the correct base using, in this case, the G as a template. to put in a new seed. However, if replication occurs before that happens, shown on the right, then we no longer have the opposite strand to use as a template. So now the polymerase does not know how to correct this. And so you can get a mutation there. Now, there are a number of different mechanisms for repairing DNA. One of them is excision repair, where if you have a mismatch, in this case a U on one strand opposite a G, you can excise that base and then add a new base to match the G. And again, if there's two good bases on either strand but they don't match, the repair process has to decide which is the right one and which is the wrong one. Now, if this occurred shortly after replication, before the new strand was methylated, it can tell the two strands apart because the nascent chain, the new one, is unmethylated, and the template strand, the old strand, is methylated. However, after methylation, then the repair process can't distinguish which one's right and which one's wrong, and will repair one to match the other, but it won't know which if it went the wrong way or the right way. Now there's another mechanism, another excision mechanism. In this case, several bases are removed that include the place where the lesion is. In this case, a repair of a pyrimidine dimer. So it's removed and chewed up, and then a polymerase can come in, use the opposite strand as a template, fill in the gap, and then ligase can seal that. So this is a number of different types of lesions that require repair. Again, a missing base, an altered base, some of the causes of these different types of mutations or lesions. Incorrect base, a deletion or insertion, pyrimidine dimer called cyclobutyl dimer in this case, or a cross-linking of the strands, which we really didn't talk about, but there are some chemicals that produce cross-links between the two strands, and of course those need to be fixed before you can replicate or transcribe through them. So generally now the structure and chemistry of DNA, double helix, make it easy to repair because we have that complementary strand that we can use to direct the repair. Now, if those strands get separated, then it's a little more difficult. There are a number of human diseases associated with DNA repair defects. We've talked about one, and that's the last one on this list, Bloom syndrome, which makes an insensitivity to UV radiation. And again, this is a defect in DNA ligase, so it doesn't hook fragments together like the Okazaki fragments. And so we saw the picture earlier of a 10 to 20-fold increase in crossing over during mitosis or meiosis in people with blooms. There's also xeroderma pigmentosa, which is a sensitivity to UV radiation. And these people have skin problems, which makes sense since they are very susceptible to sunlight, and so they generally try to stay out of the sun as much as possible. Ataxia telangiectasia, difficult one to say, is a sensitivity to X radiation, can cause lymphomas and produce a number of other symptoms. So that's all I wanted to say about repair. Again, I'll leave you with the idea that the genome does change over time. It changes slowly. There are a number of mechanisms to minimize that amount of change, but it doesn't eliminate it.